In 2005, our PI, Keith Chang, discovered that the golden mutation played the central role in the evolution of European skin color. However, Africans and East Asians have a different ancestral sequence on the gene where Europeans carry the golden mutation. East Asians, who are also light-skinned, do not carry this mutation, so they must have a different polymorphism that causes their lighter color. It turns out that this missing link is also potentially relevant to the most deadly human skin cancer, melanoma. Europeans have a 10 to 20-fold higher risk of melanoma compared to Africans. In contrast, East Asians show no increase in melanoma risk. Their rates of disease are comparable to Africans.
The goal of our project is to find the primary and genetic cellular changes in human DNA that cause East Asians to have lighter skin, but at the same time protects them from melanoma. Some East Asian skin lightening genes have been found. However, they are only carried by a percentage of the population. The mutations that we are looking for are ancient and will be carried by nearly all East Asians.
In order to demonstrate the significance of the disparity in melanoma susceptibility, let me explain how human pigmentation works in relation to melanoma. African skin is less susceptible to melanoma because the pigment protects DNA from the damaging effects of UV light. Europeans are very susceptible because their lack of pigment leaves their DNA unprotected. East Asians are also light-skinned, but are much less susceptible to melanoma. In essence, there must be something about their skin that protects their DNA.
My name is Casey and I'm a genetics researcher in the Cheng Lab at Penn State College of Medicine. I spent my graduate studies doing research among the Orang Asli, the indigenous population of Peninsular Malaysia. I became involved in this project in 2009 when I met Keith in Malaysia while he was scouting for suitable populations.
My name is Catherine, and I've been involved since 2011. I initially began assisting Casey with data collection in the States, and then accompanied him and Keith on a scouting trip to Dominica in March of this year. Casey and I returned to Dominica for a month this past June.
We need to collect samples from populations of mixed African and East Asian ancestry, where skin color can be correlated with DNA sequences. Studying this specific ancestral combination will allow us to focus on East Asian skin lightening mutations without the presence of European variants that could confound the findings. These populations will be lighter skinned than West Africans, but darker than East Asians, and only the lightest individuals will carry the primary East Asian mutations.
During Casey's graduate research, he became very familiar with one such population, the indigenous people of Peninsular Malaysia, the Orang Asli. Since 2009, he has collected 565 DNA samples and phenotypic readings from this population. In order to make sure we're identifying ancient and widespread mutations, as opposed to more recent and localized ones, we need to study another population with a similar genetic ancestry to the Orang Asli. After a global search, we found the Kalinago, the indigenous people of the Commonwealth of Dominica. Their ancestors migrated out of East Asia through the Bering Strait around 15,000 years ago. These people mixed with West Africans brought to the island during the slave trade, and the tribe developed a variety of skin colors. This created what we hope will be an ideal situation, an Amerindian tribe whose primary admixture is with darker-skinned West Africans. Once a suitable population has been identified, we collect DNA samples through either blood or saliva from individuals and families, and take a skin color reading of the right upper inner arm. In addition, we ask that everyone complete an ancestry questionnaire. Once we have collected our samples, we bring them back to our lab in the States and analyze the DNA. The next step is to test candidate genes using zebrafish, a vertebrate animal whose genes share extensive functional similarity to human genes. Many developmental and disease-specific genes have counterparts in the zebrafish.
As the world becomes more globalized, populations are becoming increasingly admixed. This makes our work more difficult because we are concentrating on ancient genetically preserved populations that meet very specific ancestral criteria. The longer we wait to complete this research, the less likely we will be able to collect enough genetically preserved samples to complete our study.
Our lab has made 3 trips to Dominica to set up the framework for our sample collection. Their council has agreed to cooperate with us, and collaboratively, we have drafted and signed a memorandum of understanding. During our last visit this past June, we spent a month in the territory, shadowing the nurses and accompanying them on home visits. Prior to that trip, we raised funds for medical equipment and supplies for the clinics serving the territory, and organized donation of a radio transmitter for the community. Through these efforts, We have put the necessary structures in place to collect 500 samples during our next visit, which at minimum will take us 2 months.
This is where your support comes in. Your contribution will fund our upcoming fieldwork in Dominica. It will cover a variety of necessary expenses, such as accommodation, air travel, car rental, food, gasoline, and compensation for field nurses. Your contribution will be tax deductible, because our lab is part of the Jake Gitlin Cancer Research Foundation and supported by the Jake Gitlin Memorial Golf Tournament.
We hope this research will provide vital clues towards understanding melanoma susceptibility. This is a chance to unravel an age-old scientific and anthropological mystery, the evolution of human skin color. If we don't do this now, we may not ever be able to.